the context of the brain at many scales, I would like to talk about the brain at a very microscopic scale, the scale of synapses and the field and results that we have been obtaining in connectomics. Now, obviously, to some degree, the human brain and any brain is a network. And the reason we are so interested in this network is because it gets built by two paths, so to say, of information storage about the world, or information about the world. One is the millions to billions of year time scale, the evolutionary time scale. And I think one can argue that this has been, of course, an important stream of information to build brains, because it has allowed us to survive. And that is even more true still today for many species whose brains are really primarily built to react to things that have been around for a very long time, like dangerous things falling from the sky, or predators approaching, or toxic substances or toxic items exuding smells and so on. But then there are the other things in our lives that actually for humans have become probably even more important in building the brain, so to say, which are the information that come into our brains during our own lifetime. And in a way, that's everything we call culture, and obviously science, all the textbooks we've read, the music we like, and whatever else, architecture and so on. So the point now is that both of these streams of information converge at the level of synaptic circuit, at the synaptic resolution connectome, not so much at projection level. And for all we know, the memories we are storing from our own life end up in synapses, right? In synapses that get built or strengthened and some removed. At least that's the prevailing hypothesis, and I would say the most likely still. And you could say that the genetic programs are mostly implemented at the level of cell types. So the idea would be that, of course, cell types are defined genetically, and rules of connectivity can be defined genetically by, for instance, recognition mechanisms. And then you build a kind of basic circuit. But again, the same circuit at a more detailed level, at the individual synapse level, starts to store information over our lifetime. And so if we understand, or if we were interested in both of these phenomena and want to understand how they converge onto the circuit, then we have to go to the synaptic level, because the individual information is likely stored at individual synapses. Now, that doesn't mean, of course, that or anyone would imply this is a single synapse, right? So no memory most likely is stored in a single synapse, but we don't even know how many synapses we need to store these kinds of experiences, right? So millions, tens of millions, 100 million, maybe even billion, maybe a billion synapses per major life event, who knows? So, but in order to find these things out, at least in the long run, we need to study the brain at synaptic scale resolution. And just a final point of introduction, this whole argument about genomic circuit encoding and individual circuit formation is, of course, extremely much species dependent. So there are clearly many animals, many species who are primarily built genetically, whose brains are primarily built from evolutionary programs, C. elegans, fruit fly even. So where essentially cell type connectivity is the key that you need to understand the kind of processing that is happening in that brain. But then when you go to mammals, for sure, that's not exclusive, of course, probably to mammals, but definitely when you go to mammals, and then even across the mammalian spectrum, you find that the configuration space of the connectome, so the number of connectomes you can build given the number of synapses and neurons that you have, explodes crazily. So this is log 10 of log 10 scale on the y-axis. So the configuration space becomes huge, and could, of course, be used to store a lot of information. The cell type based configuration space stays largely the same as far as we know, right? So the number of cell types in a fruit fly brain is, as far as we know today, on the same scale as the number of cell types in a human brain. Okay, so with this a bit lengthy introduction, I want to directly jump now to the kind of work that we do. So we do like to look at circuits at synaptic scale for the reasons given. The only method that allows us to do this, at least so far, has been electron microscopy. There are some exciting recent developments, but so far it's really for large scale and really high quality, high resolution that you need to find all synapses and all circuits data. You need the electron microscope still. So we started originally in the retina. I'm not going to talk about that today. I'm going to focus on cortex, our work in cortex, mostly mouse cortex initially as one, but then also some data from mouse versus human. And then very recently our whole cortical column connectome that I will talk about mostly because that's the most recent and for me at least most exciting thing. All right, so the field of 3D EM connectomics gained speed again in the early 2000s with Winfried Deng's technique that is shown here, the serial block phase electron microscope, where you put tissue samples into the vacuum chamber of the electron microscope and then use a scanning EM to take a picture from the top of the block and then aberrate 25 to 30 nanometers to get the next picture, and then you iterate that to get 3D data. This is was really an enabling technology at the time for our first connectomes and got us quite far. By now, most techniques use different approaches where you would first section at scale and get tens of thousands of ultra-thin sections and then use multi-beam electron microscopes that I will show later. Still, this technology allowed us to get a first highly dense and complete local connectome from the cortex. So this is here layer 4 of mouse somatosensory cortex. The blood vessels were these white large things. Now we are zooming in already. You saw the cell bodies, but now this is the beauty of the dense neuropil in the cortex. Some myelinated fibers going through, but mostly unmyelinated in the gray matter. And then you see these beautiful many small processes and synapses. So these dotted structures are, of course, the presynaptic boutons, and then for postsynaptic spine heads mostly. So this is all beautifully visible, and I still love looking at this, I have to say. This is so beautiful, and it's densely packed. This is a key point, right, to realize or to remind ourselves that the cortex is a highly densely packed neuropil where things are smushed together, so to say, to build this high-dimensional circuit that we find in our brains, where each neuron has about 1,000 other neurons as synaptic partners. So there are the cable bundles and so on. I mean, some or many of you may have seen, of course, images like this a lot, but anyway, so I always love looking at it, the cell bodies and so on. All right, good. So this was a first starting point, but now, as I already said, we are mostly using serial sectioning approaches combined with multi-beam imaging, and actually are now moving to direct section pickup on wafers, as Brigman has introduced. Okay, so this is the fast microscope that allows us to now go for a whole mouse column connectome. Many parallel scanning electron beams speed up the image acquisition, 61, now 91 beams. Therefore, we can image fast and can obtain larger data sets. We also have automated the reconstruction process so that we don't need any human proofreading anymore. This is based on AI methods. So this is a flight agent, if you want, that flies autonomously through the 3D data, very much like humans do. It's a bit like autonomous flying instead of driving. Was inspired by autonomous driving technology, and great student Martin Schmidt built this tool, and this is really replacing for us entirely the human annotation need that so far has really slowed down connectomic projects by this fully automated method. I'm just wondering whether I did switch on video mode, so maybe you're seeing. Oh yeah, no, I did not. Okay, so I just switched to video optimization. So I hope you're now seeing the video a bit better. Sorry for that. I should have done that earlier. Okay, so with these fast microscopes and the fully automated reconstruction, we can then get very large data sets. So this is our cortical column data set with about 10,000 neurons. That's important to really get at the level of an entire local cortical circuit. If you have 10 times less, then even finding all the cell types becomes difficult. But with this number, you really have the local locally complete circuitry. And this is also the largest connectome available. If by connectome you really mean all neurons times all neurons and all of their connectivity, this is what this was. So mouse somatosensory cortex, just as a reminder, and this is really not the audience where I really have to remind anyone, but is the a primary model system for the cerebral cortex, where the whiskers project obviously to the brainstem, and then thalamus cortex, 3 synapses typically very fast until the signals in the cortex, and then there we have this presumed modular structure of cortical columns. First of all, again, data from the pia up to the white matter in the bottom. It's dark here, the white matter, because of the myelination in the electron microscope. Layer 1 is clearly visible, and also already the other layers are nicely visible. Layer 4 with the small cell bodies and so on. So this is a complete data set like that, 250 microns in Z, in large scale, in XY, millimeter scale, petabyte scale, and so on. Now, then, of course, all the cell bodies, and then a volume that we presume to be a cortical column, but I'll come back to what is a cortical column later. Okay, many neurons. Now, a few images. This is now not a modeled picture where you put together cells and make it look like a column, but this is really the cortical structure as it was in one mouse. Now, importantly, I want to focus now on analyses, because also, I mean, obviously this kind of high resolution view of the brain is interesting in itself, but I do think it's important, of course, to try to really obtain hopefully relevant progress in understanding cortical circuitry and potentially even cortical function, although of course that's always a big term. So yes, there's a question. No? Pascal, you raised your hand. No, was that an artifact? Okay,
sorry, it was a mistake. Sorry.
Okay, sorry. Okay, all right. No, I'm happy to. I mean, if there's any questions or anything unclear or so, please go ahead. Okay, so so with this cortical column connectome, so first of all, what can we do? So at that scale, thousands of neurons, things like inhibitory and excitatory neurons become clear. Of course, the spine rate, as a very simple distinction, is very clear, except for layer 5, where there's a confounding excitatory neuron with low spine rate. And then we get this connectome, excitatory by excitatory, meaning thousands of excitatory neurons on both axes, and the number of synapses in this case reported as colored dots. You can also report other things here, for instance, the area of the connections as a measure of strength, but here we just use number of synapses as a starting point. And importantly, this is only sorted by depth of the soma here and nothing else, and you can already see structure. But we wanted to go one step further, meaning one step back, so to say, namely really giving up morphology. And this is something, I mean, obviously I know that, or myself, I've spent a lot of years in my life looking at cell morphology in the cortex very in much detail, and this has good reasons. I mean, obviously, as we know, it is biophysically relevant, and it is also an important distinction criterion, at least for some cells. But the question was, how informative is the connectome on its own without morphology? And by connectome, we really mean exactly that matrix here. So just the adjacency matrix of the graph, so to say, or the list of synapses. That's it. All right. So how informative is it? Well, we did something very simple. We simply clustered the connectome. Okay, the excitatory input connectome, more precisely, to all the neurons, treated as feature vectors, and simply asked, okay, what would a simple clustering? And this is, if we look at the main clusters here, then they are immediately giving us the main cortical layers. So 4, 3, 2, 5, 6 are clear immediately. Not just that, they are also, and this was really quite interesting to us, if we look at layer 4, where we know that the columnar structure, or the presumed columnar structure, the barrel structure, is very clear, then the clustering of the connectome immediately gives you this columnar structure here, where the home column is in the middle, and 3 neighboring columns are also partly there. And this was quite reassuring to us, because also, as you can see, that the location of the cells, if you look at, on the left, this z-axis plot, like linear cutoffs or so for the layers, but it's really the shape of the layers by connectivity. right? So the cortical column itself is a feature of the connectome, but also the cell types, the excitatory cell types. So for instance, we also find a layer 2 columnar structure with a center surround clustering of layer 2 cells, layer 2 pyramidal cells. Layer 6 has a columnar structure. Layer 5A has an exclusion, so to say, of the barrel, but thereby also indicating columnar structure. So this is really a feature not of morphology, at least not, we're not using morphology to detect it. Of course, you can argue that morphology is used to implement this kind of connectivity, but my main point here is that the connectome alone contains that information. And this, of course, is, I think, an important point for us, because we can now say columnar or modular structure of the cortex is a feature of the connectome. So we are, of course, now looking in other cortical areas whether we find similar structure. Now, you can also use the connectomic data to define interneuron types. This is, of course, a very complicated territory in which many approaches are available, and depending on taste, you're using one or the other. Here now is another one, which is connectomic interneuron definition. I think it's a reasonable one, because in the end circuits matter, but obviously this is also a matter of opinion. So the number we get out is about 50 interneuron types if you separate them also by layers and everything, but really just connectomically. So not explicitly saying this should belong to each other, but trying to use the connectomic features. This is actually what also transcriptomics gives you in terms of rough numbers. So maybe it's not so far from reality. Now, what can we learn? Well, first let's look at a very classical thing, the canonical circuit. As you all know, this has been a standing term, like a textbook term already, based on the very important work of Douglas and Martin, and earlier, at least statistical work or even by Breitenberg, to really ask, like, what are the features, the main features of the cortical circuitry. And for the canonical circuit, the kind of textbook picture is that you have the signals from the sensory periphery arriving in layer 4, at least for sensory cortices, and then going to supragranular layers, going to infragranular layers, and typically leaving the cortex either to other cortices or to effector channels. All right, so what about this? So here, simplified view 4, 3, 5. I mean, there are other connections, but this is really the key stream, a linear canonical circuit. Well, in the connectome, going back to the original depiction we showed earlier, you can see the canonical circuit, because pre is on the y-axis, post is on the x-axis. So you can always read it from like pre to post. That's how we like to do it. You can also transpose it, but okay. And then you have layer 4. So layer 4 as presynaptic projects to layer 2-3, and layer 2-3 projects to layer 5, right? So you see this connection. And also, if you look at the overlap of the morphology, so the axons of layer 4 go up to layer 3, and the axons of layer 3 go down to layer 5. I mean, I should say this is not fluorescence. Of course, this is actually, these are the synapses in our data, right? I'm not even plotting morphology as a feature. I'm just plotting all the input synapses and output synapses of a neuron overlaid, and then you see that. But here's the surprise. If we actually look at layer 5B pyramidal cells, so the key output or a key output neuron of the cortex, and we plot all the synapses it receives, a given pyramidal cell receives from layer 2-3, so the canonical output circuit. These are about 200 to 300, 400 maybe but then we see that the input from layer 4, spiny steroids in the same column, is almost as strong or as strong. So that was, of course, very surprising. It's true at scale. So if you plot it for all layer 5B neurons here, you see that the input from supragranular layers is similar to the input from layer 4, almost exactly. Now that is, of course, quite surprising. This means that the canonical circuit is different, that it's not serial and it's not up, down, out. It's immediately a parallel circuit where layer 4 immediately informs layer 5 before even layer 3 can do that. So layer 5 is, so to say, in full, has a full picture of what comes in way before it gets the processed output by layer 3. So how could that have been missed? in decades of work. Well, this is very interesting, or was interesting to us to think about it again. I mean, I have to say, I started as a PhD student doing paired recordings, patch clamp recordings with Bert Sackmann at the time. And this obviously, I mean, is great fun. And when you do that, you are always very happy if you get a large connection, right? You AP, action potential, and then this huge EPSP, then you're very happy. I mean, first of all, it means you get a lot of data, but also this means it's an important connection, right? I mean, a strong connection is an important connection. This was our thinking. And so if you had a connection with a unitary amplitude of, let's say, 2 millivolt, then this was a more important connection than one that had 1 millivolt, and so on. Now, this is of course true, but if you think about the circuit at scale, and for instance in a cortical column, then you quickly realize, or at least, I mean, it took me actually a moment to realize, I have to admit, but it's obvious once you know it, that of course if you want to look at the effect that a given cell population, take layer 4 or layer 2 or layer 3, has on a given postsynaptic cell, then what really matters is the number of inputs, the total number of inputs that the postsynaptic neuron receives from that population, from that presynaptic population. And this is, of course, not just the number of synapses, maybe here the size of the EPSP in a unitary connection. This is the number of synapses in the unitary connection times the prevalence of the presynaptic population, and then assuming a certain fraction of it is active. So what really matters is the product, so to say, of unitary connection strength and presynaptic population size, at least for this kind of cell type-based consideration. Well, and this is really what's going on here. The other thing that we realized is that a lot of fluorescence data can actually be misleading, because when we've directly compared fluorescent axons to EM, we found already a couple of years ago that about half of what looks like a bouton in fluorescence, examples shown here in the bottom, are not synapses. So they are either mitochondria or where's mitochondria? Here on the left, the very left one is a mitochondrion that also needs space, so you get larger, looks like a varicosity. It could be just an enlargement without a synapse. Another one with mitochondrion, and the one on the right is even a myelinated axon that can have a thickening, but obviously no synapse while it's myelinated. This is actually very interesting because it also changes our picture of the VPM innervation, because a lot of what looks like VPM innervating layer 5B is non-synaptic, is just passing by axons, but that's a separate story. All right, so I think that this combination of interpreting unitary connections as strength of a connection and the fluorescence data being at least possibly sometimes misleading has led us to not realizing this parallel circuit, and at quantitatively parallel. I'm not talking about there is a connection somewhere, sometimes between 4 and 5. That has been seen, but the point was that it's as strong, the connection is as numerous in terms of synapses. There are many other aspects to the circuit that are very interesting. Layer 2 and layer 3 have to definitely be separated, so I really urge everyone to stop saying layer 2, 3, please. It's very different. Layer 2 receives much more layer 1 input. There's even lateral inhibition between the two. Layer 3 is more combining a cell type that also gets VPM signals, but layer 2 is really, really different from layer 3, but there are also other updates to the circuit. Now, this is already quite for us in a step forward, that we now have an updated cortical canonical circuit, but we are of course very much interested in inhibition, right? Because there is so much inhibition. First of all, there's so many cell types, and well, again, I don't really have to justify it. So, so all of these excitatory spines of a mouse pyramidal cell, of course, show a dominance of excitatory input, but then there are these myriads of inhibitory neurons also projecting. So for this analysis, we took the following path. So we said, like, okay, let's look at, again, quantitatively at a typical intralayer cortical circuit. So here we're looking at a layer 3B, or so layer 3 pyramidal cell, again, just depicted in terms of all of its input synapses in black. And then we simply ask, what are all the other layer 3 pyramidal cells in the column. So we have the modular definition from the clustering. So in the column, which other layer 3 pyramidal cells innervate that particular pyramidal cell, just as an example. And there are about whatever 70 or so local pyramidal layer 3 pyramidal cells that are making synapses. And their synapses can be seen as these magenta dots on the right. And then we ask, okay, so if that is the population that can activate that or innervates that pyramidal cell in the same layer, now we can ask in the dense connectome, what other neurons do exactly these pyramidal cells innervate? And in particular, which interneurons do they innervate? So when they fire, what's the inhibition they are recruiting? Well, first of all, they are recruiting what has been well described as feed-forward inhibition by very much like basket cells, perisomatically innervating interneurons, for which we can then ask, okay, which of these interneurons are innervated by exactly this presynaptic population, and then where are those making their synapses shown in green here on the right? So perisomatic innervation. And this is really very much as we expected. This is cellular feed-forward inhibition, as we call it, because it's really cellular precision. It's not just average connectivity. It's really each of these connections are a triad, so to say. So E to I to E, and the same E goes to also the same E. So this is a triadic feed-forward inhibition. Now, to our surprise, we of course searched for other interneurons, or asked, is that the only interneuron that's doing that? To our surprise, there are more. So how do we analyze this? Well, we say that, okay, let's take all EE connections in this layer and ask for individual other interneurons whether they are closing the triad, so to say. So do they convert E to E into EIE in each particular case? And so here this diagram shows that whenever there is such a triad, then there is a black line. So you can immediately see that this interneuron type, whatever it is really systematically closing the dyads into triads. So we can quantify this by saying, okay, what is the percentage of EE connections that is covered by that interneuron? So that's a coverage. And also we can ask how many interneurons of a given interneuron type provide this. And this is the mean or median or whatever number of CFFI triads per EE pair. And now if we take those two measures, then and plot this for all interneuron types in our column, we see that in fact, as expected, the feed-forward inhibition by perisomatic innervating interneurons shown in green there is the most prevalent. It's covering almost each and every pair, and with 5 to 6 interneurons per pair, so very effective. But there's a second interneuron type that also has very high coverage, and this is actually an interneuron type that you would call distally innervating, because it makes its synapses also on the layer 3 pyramidal cell, but not around the soma, but out there where the excitatory synapses are being made, so distally innervating. Now, this is very important. Again, quantitatively, these are as many synapses, and as you can see, they are actually placed exactly in a position where the excitatory synapses are coming in. So in other words, what we find is that if you naively look at the circuit without any assumptions, and ask which inhibition is recruited effectively and effectively and also with substantial impact, then we find 2 channels of forward inhibition in the column and not just one. And the distal inhibition is as efficiently recruited as the proximal inhibition. So this, of course, now asks for how can you actually get any signal through, because if you have as many inhibitory synapses being activated in the same presynaptic population via the triad as the EE connection directly provides, then this is like an IE balance, effective IE balance of 50-50, meaning you have no chance to get your excitation through, especially if you also have speeding up inhibition. So here, of course, now disinhibition comes in. So we then looked at which interneurons could stop this forward inhibition. And these are the well-known bipolar type interneurons, probably VIP-like interneurons, And again, we can ask, do they make enough synapses onto given interneurons to actually shut them down? Here we are not talking about 40, 50, but rather about 20, 30. But plausibly, biophysically, this is already able to stop this interneuron. And here the next surprise was that these disinhibitory interneurons are routinely activated feed forward. So in the textbook, or well, maybe not yet textbook, but in most circuit diagrams, you see the bipolar VIP type interneurons are thought to be top-down innervated and maybe provide contextual modulation of signal flow and so on, or even unlocking of local excitation in an attention idea also. But we find that half of the bipolar interneurons, roughly, so half of the disinhibitory interneurons are activated bottom up. So the thalamus already innervates one type, then layer 4 innervates another type, and so on. So essentially, the picture that we think we are seeing here is that we have a circuit where EE connectivity is extremely strongly inhibited away, so to say, by two-channel 50-50 IE balance inhibition, and that then, in order to get any signal through, you need to disinhibit the circuit, and that is being done feed forward, so that you essentially switch on your gating or switch on your path, your transmission path. Okay, so these are two example analyses that I find currently most important. We are of course looking also at other top-down input combinations with bottom-up and so on. One other thing that we're also very much interested in is to simulate, of course, some effects you can study, back of the envelope, but of course with this complicated circuitry, you need to at some point go into the details of the transmission delays and so on. So we are happy to also simulate this and have now managed, or this is a student, Apurva, managed to get all of these thousands of neurons excitable, and thereby we can now move into a more systematic signal processing study. So I'm just looking at the time. I have a few more minutes, or should I stop already? 45.
Yeah, you can continue.
A few more minutes? Okay, all right. Good. So I think, so we also, of course, analyzing the top-down input, how it combines with bottom-up input in the connectome, right? I mean, this is not a functional measurement. This is just circuit analysis in detail. You can ask, for instance, where do the two streams of information ideally converge? And so far, it turns out that lower layer 3 is an ideal convergence point, but this still needs to be worked out more. All right, so I really made sure to focus on the new column data. We also, of course, looked at the potential traces of Hebbian plasticity in the column, which we had originally done based on the synapse analyses in our 2019 paper. Cut a long story short, Hebbian learning makes a prediction about pairs of synapses to get stronger together, and this is something you can quantitatively observe. And we have now also conducted this in the column data, allowing us to ask, are there any circuits in the column that seem to be more prone to Hebbian plasticity, to cut it short? I mean, there are a few caveats there, of course, but so what I just want to emphasize here is the signature that we find that layer 5, particularly layer 5B, the incoming connectors to layer 5B, at least those from supragranular layers, are at least twice or almost, and sometimes more, twice as strongly in this state that we would interpret as a possible Hebbian state than in any other connection in the column. So it's interesting to see that this could be a site of enhanced plasticity. Interestingly, the shortcut connection, the 4 to 5, doesn't show this signature so much. So there might be a very interesting crosstalk here between linkage connections or storage connections and just signal processing connections. All right, so these are just visualizations of the same. All right, so this briefly summarizes the key points from the column. So where I already said, I think the updates to the circuitry. I think I'm not going to talk about our mouse to human comparison today, which we have already published a while ago, where we find, but that closes loop. So let me just briefly mention this. So we find that in human, really the most important change in the connectome compared to mouse relates to inhibition. We have 3 times more interneurons. They are not the same kind as in mouse, in the sense that it is exactly those bipolar VIP-like interneurons that are substantially expanded. So half of our interneurons are of this bipolar type. And this is, so we have 3 times more interneurons than mouse, and half of our interneurons are bipolar compared to the mouse, where it's only 20% of the 12% interneurons. And our interneurons, meaning primate interneurons of this type, of this bipolar type, are able to innervate other interneurons exclusively. So really 100% of their synapses are made onto other interneurons, meaning this eye-eye connection that I talked about earlier in mouse already, which we think in mouse already may be needed to unlock signal flow in the column, is now exactly the connection that is not just subtly but tenfold expanded from mouse to human. So almost tenfold expansion of this eye-eye connectivity. And this is, of course, quite interesting to us to follow up on. First of all, is it really a mouse to primate peculiarity, or what about other species? So we're looking into that. But also to think about what this could mean. There's one interesting theoretical prediction I don't have here from Sinofsky's lab that talks about timescales, where eye-eye connectivity is linked to a prolongation of timescales of percept maintenance. This is, of course, a very attractive interpretation that maybe more interneurons of this kind might contribute to us being able to keep our thoughts for longer. But that is, of course, a very far-reaching interpretation. All right. Yeah, I mean, obviously in the field we're very ambitious. We want to go for larger brains. The question is, will it take a few years or many, many decades? This depends a lot on funding and scaling and the degree to which the AI world is interested. But again, I try to show that really for cortical circuit analysis, we have a lot that we can still discover. This will be even better when we can screen. So this is the goal of screening connectomes, not with 1 or 2, but with hundreds to thousands of samples to discover pathologies and also look at behavioral correlates and so on. And this we have made possible recently by combining simply many, many samples into one and then very efficiently imaging them, so that we are really getting to the hundreds to many hundreds screening scale with this kind of multiplexing approach. And that will also allow us to look at the temporal processes in more precision. All right, with that, I think I'm still just in time. Thank you very much, and I look forward to your questions.
Thank you so much, Moritz. Do you see an image of the room? Not now, maybe. Or can you see the audience?
Now I see the room. Yes, hello. Nice to see you.
Okay. So questions. Yes, start.
Thank you for your talk. Wait, should I look this way? Oh.
I mean, you see him better that way, but I guess he sees you better.
Thank you for your talk. I was wondering, in your data set, do you have axons coming from different cortical columns and different areas, and how do you differentiate those connectivities and how they are like local column connectivity versus multi like long range aerial connectivities?
Yeah, so I mean, obviously, so the volume is just large enough for a column. Actually, it is containing several columns, so we're now looking into cross-columnar circuitry as well. In terms of other cortices, this data set is not yet large enough. So step by step, yes. So now we offer, of course, also going for data sets that do actually contain, for instance, S1, S2, or so, that we can actually look at the connectivity at high resolution. Now, in our data, what we do have is we do see these layer 1 axons coming in. We don't know exactly, of course, where they're coming from, but we can at least analyze in principle what kind of circuits they trigger in the column, assuming that they are long or like top down, mostly at least, or to some degree. But yeah, so of course in the future the ideal situation would be if we have everything in one data set. and then we can analyze also the long range at high precision.
Thank you very much.
Moritz, I had a question about the similarity of connections between neighboring neurons. So this microcircuit that you referred to, like if there are two neighboring excitatory neurons, are they going to share this microcircuit, canonical microcircuit, or they'll make their own?
You mean like individual postsynaptic neurons, whether they how different? Yeah. Good question. So I'm just thinking, what have we analyzed in that direction? So I mean, we have so far, I have to say, treated the. So after seeing these modules, these clusters in the connectome of the layers and so on, we've then treated all cells in these clusters as rather homogeneous. But that is of course not necessarily true. So I don't think we have looked at geometry or like location dependence, if you say neighboring location dependence, beyond these connectome clusters at this point. So that's an interesting point. No, I haven't. I mean, they share. I mean, I think they do usually share, but it's not. I mean, 70 out of hundreds. It's not a huge fraction. So obviously the connectivity is not very high. So it's sparse enough to have selectivity. But yeah, no, I don't have any data right now. But this would be easy to analyze. Thank you.
Yep.
So the VPM axons actually building the column, do they connect to all layer 4 neurons or only to a subset? There are some recent suggestions that they only connect to a subset. What is your measure here?
So again, I think, I mean, we have analyzed it, and it's the vast majority. I mean, maybe there are a few, at least, I would say 90% definitely. So are contacted by VPM. So maybe there are 10%, but we haven't looked for these. What is very interesting is that the VPM input that was always presumed to also go to or also target layer 5 in layer 5, right? Because there are these branchings there, that this is actually mostly non-synaptic. Yeah, so this is mostly just branching. So but that's targeting layer 5, not your question. Your question was layer 4. What we also found, which was very interesting, and is I think consolidating now in the data, is that this VPM channel seems to consist of two types of axons. One that is really layer 4 only, and the other that also goes to layer 3. So this seems to also separate.
I have an additional question. You mentioned the layer 1, and you mentioned that there are some connections to pyramidal neurons layer 3 and 5, but there should also be connections to inhibitory neurons. The connections to inhibitory neurons more either per synapse to the interneuron or connecting to more interneurons like Martinotti's and others.
No, and quantitatively they are not more. They are not so many. The vast majority, and this is my main reason to also emphasize this distinction, the vast majority goes to layer 2 pyramidal cells. Yeah, and this is really because they are so close, and they even have most of the apical obliquely reaching into layer 1. So they have like hundreds of synapses from these inputs, or even reaching 1,000 and more, compared to, for instance, layer 5, which is much less. So the fact that they have so massive dendritic part of the dendrite in layer 1 means they are the key recipient of whatever comes in up there. And that's interesting because you can then study how this could trickle down into the column. And this is both by a direct connection to layer 5, of course, but then also via layer 3. But the interneurons are targeted, just to come back to your question, they are targeted, but quantitatively that is not dominating the circuitry. And interestingly, for instance, we find, I should say we are still looking at it, but it looks like there's also interesting types of inhibition excitation circuits triggered now by these axons, right? Where that could innervate interneurons and excitatory neurons, and this is something that we're also looking at.
I think we had at least 2 more questions. We're running a bit over time, but I think since Moritz isn't here, it's better to take those questions as well. Yeah. So there.
So, Moritz, super interesting, really fascinating finding. In particular, this relative enrichment of the human cortex of these inhibitory motifs. Do you have questions? Is it area specific? For example, more prefrontal specific? One question. Second, is it possible from a data set to have an insight of which is the main input source of these sort of extra cells, like cortical versus neuromodulatory, for example?
For the bipolar type interneurons, you mean?
Yeah.
Okay, so first question, cortical areas. So, I mean, there is some report that there is a frontal dorsal axis in terms of I to E ratio, and so in general, the thinking is that the frontal brain, so to say, or the more frontal brains, parts of the brain, maybe have more of this. This is, however, a subtle effect, and the effect that we find is like threefold change. We checked by looking at multiple cortical regions in mouse, at least. It doesn't seem to be a major determinant. It could be that other species have a stronger difference between prefrontal and frontal regions, if they exist, and more posterior ones. So all I can say at this point is that in human we looked at a frontal and a temporal gyrus, in macaque at S1 and temporal, and in mouse at many areas, and this effect was absolutely dominated by species dependency. So primates, a lot of II, 3 times more interneurons, and mouse, subtle changes, but nothing at that scale. About the input, yeah, so I mean that was partly what I tried to, but I only summarized it. I didn't really show the data. So we asked exactly that question. So what are the main inputs that could switch this bipolar circuitry on? And we found that for half of them, not all, half of them roughly, the main input is from VPM, layer 4, and layer 3. So meaning that you have types of these bipolar interneurons that could really be used to switch on your forward excitatory channel, so to say, by disinhibiting the inhibitory systems.